Today I am going to discuss with you all about the micropropagation. Now, micropropagation is also called as clonal propagation. So, as definition given here, micropropagation is the asexual. It is an asexual or vegetative propagation of the whole plants by using tissue culture technique. So here we will do in vitro propagation of plants. Okay, we will propagate the plants, mainly the vegetative part of the plant. The vegetative part of the plant includes your leaves, roots, leaf buds, and stem, right? Although they will not directly involve in seed production, but these vegetative parts can often be used in vitro. Sometimes they are also used in vivo in certain plant species like banana and grape. Sometimes we also use the rose plant also. We use the stalk of the rose plant to generate the stem of the rose plant. So in vivo we can do with certain plants by taking their vegetative parts for propagating. In vitro, we are using these parts for generation of whole plants, and that's why it is also called as vegetative propagation. So plants can be plants can be propagated by sexual means, that is through generation of seeds, and at the same time they can also be generated through asexually by multiplication of the vegetative parts of the plant. Now, this micropropagation, also called as clonal propagation, which refers to the process of asexual reproduction by multiplication of your genetically identical copies of individual plants. So the plants that we will get through micropropagation process, they will be genetically alike plants, just like the mother plants from where the explant has been taken from. Asexual reproduction through multiplication of vegetative parts is the only method for in vivo production of certain plants, as they do not produce viable seeds. Now, there are certain plants which do not produce viable seeds. Viable seeds means the ability of the seed to germinate into a new plant. That is the viability of a seed. Now, some plant they cannot able to produce viable seeds like banana, like grape. We have not seen a seedless. We have seen seedless banana, but there are also seed banana. But that seed will not generate into a banana plant, as it is sterile. It is non-viable. Normally, there are here some ways that affect the seed viability, and we can check the viability of the seeds by a simple test. We can also use biochemical test. Like for example, the water test is one of the tests where we can observe that whether the seed is active, whether the seed is viable or not. Just we have to place the seeds in a container of water and let the seeds soak in the water for 15 minutes. If the seeds sink to the bottom, they are viable. That means the water is entering to their through the minute pores in the mouth where the embryo development will be occurring. There is a particular name in that part. And if the seeds float on the water, that means they are not viable. Various factors affect the seed viability, like seed moisture content, temperature, relative humidity, initial seed quality, These are the factors that affect the seed viability. So many plants, they cannot able to generate, their seeds are not viable. So micropropagation is a good way that we can use those plants for those commercially important plant or plant of medicinal value for generating millions and millions of plants by using their vegetative part of the plant as a explant. Clonal propagation, that means micropropagation, has been successfully applied in apple, potato, all the tuberous plants, and several ornamental plants, which are the plants that are normally grown in the garden for beautification only. Now, there are certain advantages of this micropropagation. Faster multiplication. First, very quickly we can generate into many plants. Large number of plants can be produced from single individual in a short period of time and space, irrespective of seasonal variation, whether it is summer or winter or monsoon. We can grow the plants in vitro within the lab in a controlled culture conditions, free from pathogens. So many plants can be created. Second is possible to produce genetically identical plants. Now, genetically identical means the genotype of the plants that we are taking from and the plants that we have generated from by taking the explant from that mother plant will be same. Now, depends on from where which tissues we are taking. Normally, meristematic tissues are the best source to produce genetically alike plants. As I have said in the previous videos, that meristematic tissues, they divide by normal mitosis and the cells are maintained in diploid level, and they are very actively dividing cells, so there is no scope of entry of pathogens to carry out their replication, to carry out their copy number. That's why meristematic parts are good way for creating a disease free plant. Second is, if when we are talking about genetically identical plants, the non-meristematic plants, they don't divide by normal mitosis. The cell enter to the endoreduplication that leads to the formation of chromosome with 4 chromatids, 8 chromatids, and so on. That's why meristematic tissues are taken, so that they are normally actively dividing meristematic cells that maintain in diploid level. They go through more normal mitosis. So it is possible to generate your genetically identical plants. Second is sexually derived sterile hybrids can be propagated. Now, those which are hybrids that we have crossed from one plant species with the other plant species of the same genera, maybe to produce a hybrid plant, but that hybrid seeds that we obtain are not compactable, they are not sterile, they are sterile, that means they are not able to produce new plant. So that type of sexually derived sterile hybrids can be used for micropropagation for raising more and more hybrid plants. Seed raised plants pass through an undesirable juvenile phase which is avoided in sexual propagation. Now this you can say as off season plants. It is referring to as juvenile phase. Juvenile phase is a period of time when plants are not capable of flowering and all organism has to attain certain stage of growth and maturity before they can reproduce. So immediately the plant cannot able to reproduce. For this they have to grow at certain stage of time, they have to mature, they have to mature their reproductive part of the plant, then only they can able to do pollination. So that phase where the plants are preparing, they have to attain certain stage of growth and maturity before they can reproduce. That phase is called juvenile phase. So that type of juvenile phase can be overcome by using vegetative propagation. You can say indirectly, forcefully, we are trying to grow the plant in a quick period of time. We are providing growth, and at the same time we are overcoming this juvenile phase of the plant. The last one, it is saying that gene banks can be more easily established by clonal propagated plants. What are gene banks? Gene banks are a type of biorepository which preserves the genetic material, right? So it is just a type of, just like an account. In bank account we actually store our money. So gene bank are the type of biorepository where we are storing the preserving the genetic material of many species, especially endangered species or species of a clinical importance. Okay, a collection of seed, plants, tissues, tissue cultures are potentially useful for containing the gene of significance, and they are generally stored and preserved in gene banks. Now, in many ways, the gene banks are used to preserve the genetic material. One way is by cryopreservation. Cryopreservation is a process where the tissues, the plant tissues mainly, are first treated with certain chemicals called cryoprotectant, okay, which normally stabilize their internal environment. They are internal, so that they that does not get freeze. The internal environment does not get freeze, and at the same time we will put them in liquid nitrogen at minus 196 degrees Celsius to store them for a long period of time. So that is also a way to preserve our genetic sources. Now in India, a gene bank is located in Karnal, Haryana, and the name of the gene bank is National Bureau of Animal Genetic Resource, NBAGR. Short form is NBAGR, National Bureau of Animal Genetic Resources. Now, this micropropagation technique has been first started by Morrell in the year 1960. So you can see Morrell in the year 1960 have first started this technique, and he have used in orchid plant, okay, which is a type of ornamental plant, and now applied to several plants of commercial importance. Micropropagation is a handy technique and it is used for rapid multiplication of plants. So let us now move to the next part, which is the technique of micropropagation. Now this technique of micropropagation, as you can see, micropropagation consists of 5 stages. There are 5 stages in micropropagation, ranging from stage 0 up to stage 5, sorry, stage 4, up to stage 4 is there. Okay, so let's have a start now. Okay, so first is stage 1. Your first stage is stage 1. Sorry, stage 0. Your first stage is stage 0. So in this stage 0, what it says, in stage 0, what it states that stage 0 or this stage involves selection and growth of stock plants for about 3 months under controlled culture conditions. So in this stage, the plants from where we want to take explant from it and culture them in a suitable culture media, that mother plants are selected and they are grown in controlled culture conditions for 3 months, and it is mainly done why not to prevent the plant being affected by pathogens, so that we can obtain the explant from the plant which is free from pathogens. And we're actually trying to adjust the environment of that plant so that it can grow well, so that that young part, the newly formed part of the plant, are generally used as explant, which are actively dividing growing cells, especially the meristematic regions like apical meristems, then the axillary bud, which is also rich in meristems, meristematic tissues, right? Just beneath, just after the root tip, just before the root tip, there is also a meristematic regions that can also be used to generate callus. Okay, so the first step in micropropagation is we have to properly select and grow the stock plants, the plants from where we are going to take that explant in a controlled culture conditions, to as to prevent the so that the plants does not get affected with pathogens and it can grow well. The stage one, that we can see that stage one is the stage where we initiate and establish the culture in a suitable medium. The stage one is the stage where we are all doing all the stuffs. We are sterilizing the equipments, we are sterilizing the explants, surface sterilization. Then we are within the laminar airflow, we are inoculating the explants in a suitable liquid, in a suitable medium, aseptically. Then we store them in the culture rooms for their growth. So in stage one, it is the stage of initiation and establishment of culture in a suitable medium is achieved. Selection of appropriate explant is very important. Yes. So explant selection is very, very important, that what type of explant you are going to take. And most commonly used explants that we normally use for micropropagation is your shoot tip and axillary bud. Okay, so what are shoot tip, what are axillary bud, you all know. So this is the shoot tip. If I show you in the diagram, so this is a scalular apical meristem, and you can see what is axillary bud, just the stem and the stalk, and you can get that axillary bud. Okay, so from these regions, sometimes we also take the internode regions, the nodal segments, which are node and internode region, can also be taken. Okay, and directly you can also take that axillary bud, which has just arise from the petiole of the leaf. Okay, so generally the shoot tips or axillary bud, what we are taking for generation of micropropagation for plants, they should be surface sterilized and washed before use. Now, normally the explants that have meristematic tissues are generally used for micropropagation. Meristematic tissues are found in many locations, like they are found in the tip of the roots and stems, they are found in the buds, in the nodes of stem, in the cambium between the xylem and phloem. Okay, especially in dicotyledonous plants. That's why we prefer to choose that regions, the shoot tip, the root tips, okay, buds, nodes of the stem, cambium between the xylem and phloem. Okay, now why we are taking this meristematic tissue as explant for micropropagation, as I have said, meristematic tissue have continuously and rapidly dividing cells, and these cells have a high rate of division, high rate of metabolism also, and thus pathogens like viruses cannot able to replicate inside their cells, because they are dividing, so there is no scope of entry and carry out their propagation, carry out their division. And most of the pathogens migrate through the vascular elements like xylem and phloem, which are the conducting tissues, right? But in the meristem region or the shoot tip region, there is barely your xylem and phloem is there, so there is no chance of entry of virus or pathogens in the meristematic regions. In the meristem region, that is in the shoot tip, also the tip of the plant, there is a high concentration of oxygen also, that decrease the growth of pathogens. Okay, and at the same time, I have said that meristematic explants have a high degree of sorry, not high degree, sorry for that. Meristematic explants have less degree of genetic variability as compared to the non-meristematic explants, and the reason is the cells of the meristematic explants, they divide by normal mitosis, and the cells are maintained at uniformly diploid level. So as the cells are constantly dividing by normal mitosis and the cells are maintained at diploid level, so we can obtain genetically similar plants from the mother plants. While the non-meristematic explant, they don't divide by normal mitosis, but they undergo endoreduplication. And endoreduplication is a process where DNA is replicating, it is making its copy, but the cell division does not occur, the cytokinesis does not occur. Okay, and thus it leads to the formation of chromosome having 4 chromatids, 8 chromatids, polychromosomes, etc. So that's why we take the meristematic tissues of the explant as it is free from virus. Second, it maintained in a uniformly diploid level, so we can get true to type or genetically similar plants from the mother plant. Let us see now the stage number 2 now. So we have already prepared the culture. Okay, we have already prepared the culture. So in stage 2, as you can see in this stage, the major activity of micropropagation occurs in a defined culture medium. Stage 2 mainly involves multiplication of the shoots or rapid embryo formation from the explant. So in the stage 2, normally what we do subculturing of the plants that has been cultured, that has been stored in the culture room. Okay, now the main purpose of micropropagation is formation of plants. So we use a defined culture medium. Normally the culture medium that we use is Murasi's and Skoog medium. Sometimes White's medium, Gamborg medium can also be used. So normally in stage 2 what we do, we change a little bit composition of the culture medium so that our target, that is multiplication of shoots, can occur, or embryo formation can occur from that somatic cells of the explant. Now for the embryo formation, this process is known as somatic embryogenesis, where we develop somatic embryo from the plant. And stage 2 involves normally multiplication of shoots or rapid embryo formation from the explant. Okay, so normally we know all of us that we have to incubate the culture vessel and culture room at 25 plus minus 2 degrees Celsius at 40 to 50% humidity. Lux, the light intensity will be 2000 to 3000 lux light intensity for 16 hours photo period. That means 16-hour light phase and the rest 8 hours dark phase for 2 to 4 weeks. We have to monitor them at each week or after 3 to 4 days. Okay, so that any contamination occur in the medium or not. So the stage 2 mainly involves multiplication of shoots or rapid embryo formation, that is shooting, you can say. Then comes the stage 3, where in this stage it involves the transfer of shoots to a medium for rapid development of roots. Now after shooting, multiple shoot formation occur, they get their achievement height. Now we have to do rooting of the shoots. Okay, so that we can later on we can, they depend on their own then. Okay, they will not depend on the culture medium then, because at in the next stage we have to put that plantlets into greenhouse where they have to obtain their minerals and nutrients from their own with the help of roots. So in this stage we are transferring the shoots into such medium which will develop into roots, which will develop root development will occur. Now, sometimes the shoots are directly planted in soil to develop roots, like in case of banana also. We directly after shooting, we do not use a medium for root development. We directly place them in the greenhouse, which is also called as hardening for rooting formation. Next is stage 4, which is the last stage, and this is the main stage where the plant has to get exposed to various stress conditions, because we are now placing the plant from comfortable zone to a stress zone where the plants now itself have to obtain their nutrients from the soil using their roots that has been developed in stage 3. So this stage involves establishment of plantlets in the soil, and it is done by transferring the plantlets of stage 3 from the laboratory to the environment of greenhouse. Okay, now normally in the greenhouse the plant species are actually planted in pots that have a suitable compost mixture, and that suitable compost mixture I have already discussed with you little bit in your first lecture. It should have a ratio of sand, sterile soil, and manure, which is also called as farm yard manure, made from the excreta of animals in a ratio of 1 is to 1 is to 1. So sand, sterile soil mixture, and manure together they are mixed in a 1 is to 1 is to 1 ratio, and this mixture is used in the pots for growth of these plantlets that we have taken out from the laboratory from stage 3. Sometimes instead of using sterilized soil, we can also use soil right, cocoa peat, or perlite for their growth. Right. Now, micropropagation is a lengthy process, as you can see, but it is not so much lengthy if you compare with the growth of the plants normally in vivo condition, because in micropropagation, if you see the micropropagation process, it is lengthy, okay, but within that time we can produce many plants which are not possible naturally in vivo technique. Now it is saying that micropropagation mainly involves in vitro clonal propagation by two approaches. One is multiplication of axillary buds, that is, we will use the axillary buds or shoot tips to grow into plantlets. And second way of micropropagation is multiplication by adventitious shoots that have the nodal segment, normally Y-shaped nodal segment, just like this. Uh, Suppose this is the, yeah. So this is the axillary bud that you can see. Suppose this is the axillary bud. Okay, so this part we will use. Okay, the leaf will be eliminated because it will take the most of the nutrients. So we will make them just like this. Okay, only the Y shape. We have to cut also this one to make a little exposed part like this, like eggplant. Okay, so the adventitious shoots are also being used for micropropagation. Okay, so besides this micropropagation, there are also other two processes which are also involved in micropropagation, like organogenesis and somatic embryogenesis. Organogenesis, as the name suggests, is actually we are forming individual organs. Okay, like we are making organs like shoots, roots directly from the explant. Okay, or from the callus and cell culture induced from the explant. So Organogenesis is a process where we are directly form the organs, plant organs like shoots, roots directly from an explant or via with the help of callus, which has been established in the culture. The somatic embryogenesis, as I have mentioned a little bit, it is a process where the embryo development will occur from the somatic cells of the explant. Now, this is a very interesting technique where we are developing embryos in the somatic cells by just changing the composition of the nutrients, hormones in the culture medium, and that will leads to the development of embryos in the shoots. And then these embryos are taken out and they are coated with sodium alginate calcium chloride solution to make a polymer of to make a polymer which is encapsulated within the which is encapsulated around the embryo, and that is the artificial seeds. These seeds now can be used as normal seeds, treated as normal seeds for production of plants in vivo condition. So we will both also go through this organogenesis and somatic embryogenesis process also later on. So let us see the multiplication by axillary bud and apical shoots. Now it is saying that what is axillary bud actually? Axillary bud is an embryonic and organogenic shoot which is located on in the axil of a leaf. So I have mentioned with you about the axillary bud. If you see the axillary bud, okay. So this is the axillary bud, okay. So you can see the node, the nodal segment, the internode, okay, and just after the petiole that joins the leaf, the base where the petiole has been joined with the uh, main stem of the plant, that is the axillary bud located, and is also a more region of meristematic tissue, so we can use them for the growth of for micropropagation process. So it is saying that quiescent or actively dividing meristems are present in the axillary bud and apical shoots, which are also called as shoot tips. So we can the actively dividing meristems are present in the axillary bud and in the apical shoots. Axillary buds are located in the axil of the leaves, as I have shown to you just now, and they are capable of developing into shoots. So these axillary buds are have the capacity to grow into shoots. So in vivo conditions, there are limited number of axillary meristem that can form shoots. Some of the plant species can use this axillary bud in vivo to form shoots like rose plant. By means of induced in vitro multiplication, it is in micropropagation, it is possible to develop plants from meristems and shoot tip cultures and from bud cultures. So let us see that how these axillary buds and the apical shoots can be used for micropropagation. It is saying that apical meristem is the home, or you can say dome, of tissues located at the extreme uh, tip of the shoot. The apical meristem along with the young leaf primordia, which will give rise to leaves that will constitute the shoot apex. So it's just look like this. If I say it just look like this. So if this is the apical shoot, this is the leaf primordia, this is the apical shoots, these are the leaf primordia, these are the leaf primordia. It look just look like this. Okay, so apical meristem is the home of the tissues which are located at the tip of the shoot, and this apical meristem along with young leaf primordia made the shoot apex. Now, for development of disease-free plant, meristem tips should be cultured. I have already said they are free from conducting tissues like xylem and phloem, so there is no scope of entry of viruses or other pathogens. Second point is that they are actively dividing growing cells. They are very metabolically active cells, so that there will be no chance for the pathogens to get into it and carry out their cell division. Okay, so normally the meristem or shoot tip is isolated from a stem by a V-shaped cut. So in the V-shaped cut, as you know, it will look like both way cut. Okay, just in the meristem tip. So we take the meristem tip, we make a V-shaped cut, and the size should be zero point 0.2 to zero point 0.5 millimeter. That is critical for culture. Now it is saying that larger the explant, better the chance of culture survival, but at the same time, if we are taking a larger explant, that becomes a problematic because we are taking also the non-meristematic parts of the plant if we are taking larger explant. And you know that non-meristematic parts have the conducting tissues, and there's a chance of having pathogens. Okay, so all the positive and negative points are there if you are taking larger explant. Even if you are taking smaller explant, there is a less chance of survival of that explant. That's also a bad point. So for good results, we have to take zero point 0.2 to zero point 0.5 millimeter of the tip, and explant should be taken from actively dividing growing shoot tips, and also the ideal timing at the end of the plant dormancy period. Now every plant have a dormancy period where the plants does not grow. Okay, at particular time the plants grow. Like you can see that all the plants, the evergreen forests are different, they grow all the time, but the deciduous forest trees, plants, they at the particular season they all shed their leaves, right? And at the spring season their leaves are generating flower, they bloom their flowers, right? So we have to take the explants during that period, during when the dormancy period of the plants has been ended. Okay, and the most commonly used media for meristem culture is your ms medium and whites medium. MS medium is made up of various type of macro elements, micro elements, vitamin source, amino acid source that have a particular composition depending on what amount of culture you are using to, and what type of explant we are using for this medium for their growth. Okay, now there is a diagrammatic presentation that has been said, that has been mentioned in the show, in this shoot tip culture or mainstream tip culture, that has been shown. So I will show you in the next page that diagram. So in stage one, the culture of meristem has been established. So in stage one, the culture of meristem has been established. Addition of growth regulators like cytokinins, for example of cytokinins kinetin, then BA baric acid, and some auxins like naphthalene, alpha naphthalene acetic acid, indole butyric acid can be used, that will be helping for their growth and development. So these are used in the culture medium so that the plants can able to obtain that nutrients and grow in the culture medium. So in stage one is the all stage where we are growing the explants we are growing the explants with suitable growth regulators like auxins and cytokinins in a suitable ratio for that shoot development will occur from that meristem tips. So this is the stage one. I am zooming it. So this is the stage one. The shoot tip or the meristem is taken from the shoot apex of the plant and it is poured into a culture medium having a good condition of your concentration of your oxygen and cytokinin in a suitable ratio, right? Next comes the stage 2. If you see the stage 2, this is the secondary culture. In stage 2, what happens? Shoot development along with auxiliary shoot proliferation occurs. So in stage 2, you can see shoot development along with axillary shoot formation, shoot proliferation occurs. Now, so axillary shoots or the helper shoots grows. So multiple shoot development occurs, and for this we have to change bit of composition of the culture medium. We have to add high levels of cytokinin to enhance the shoot proliferation. As we have studied before, that high cytokinin, less oxygen is desirable for shoot proliferation. Then obviously in stage 3 there is rooting. In rooting stage, we know it is facilitated by low cytokinin and high oxygen. You can see the stage 3, that for rooting process, high levels of oxygen and less amount concentration of cytokinin is preferred, which is opposite to the shoot formation. Okay, so obviously you can see that the for shooting purpose at stage 2, the composition of the medium is bit of different from the stage 3, where the routing of the culture media has been done. Okay, so repeatedly we have to do subculture, and that's why it is a bit of lengthy process. But at the same time, we are also seeing the positive side, that we can grow many plantlets. We're not using one explant to grow into this plant. We are taking millions and millions of explants for growing into those such type of plants. Okay, and the original temperature and the culture range should be within 20 to 28 degrees Celsius, and majority 24 to 26 degrees Celsius is preferable. Lower light intensity is more appropriate for good micropropagation. So this was about the meristem culture, the shoot tip culture, where we can see the shoot apex from the plant has been taken. It is properly surface sterilized and inoculated in the culture medium, having a desirable composition of the culture so that they can grow into shoots. Okay, they can grow into callas, and from the callas they will grow into shoots. Now shoot formation requires a bit of change in composition of the culture medium. High cytokinin, low oxygen concentration is desirable for shoot proliferation. For the root proliferation, the opposite thing will occur. Change in the composition of the medium, low cytokinin, high oxygen concentration proliferates root formation, and thus we make a rooted plant. Okay, and thus then we will, after desirable, the plant get desirable increase in the amount of their adventitious shoots and roots. Then we will put them in a greenhouse in a pot mixture of sand, sterile sand, soil, and your farmyard manure in 1:1:1 ratio. Okay, next is your auxiliary bird culture. Now auxiliary bird are those plant birds which possesses quiescent or active meristems. Quiescent means cells which do not divide, but they retain their ability to re-enter cell proliferation. So they don't divide, but they have the ability to re-enter into cell proliferation stage. That's why we have said that each of the cells in the plant tissues are totipotent, have the capability to give rise into a new plant. We have to make change in the culture composition for growing that plants, but they have that capability, just like your auxiliary bird. They are quiescent, but they can divide. They have that ability, they can retain their ability so that they can proliferate, they can do cell proliferation. Now two types of bud cultures are used. One is single node culture, where we are using the entire node, nodal segment of the plant along with the auxiliary bud, and there is auxiliary bud culture where only and only we are using the auxiliary bud. Okay, so single node culture is a natural method for vegetative propagation where we are taking the nodal segment just like this. I have already drawn just like this the nodal segment, the internode segment, and also the bud, also this bud, that bud, auxiliary bud that we can see. So whole thing we will use as explained. Also we will cut them into smaller piece to look like this, to look like this. Okay, and use them as explained. Okay, so it is a natural method for vegetative propagation of plants in both in vivo and in vitro condition. The buds found in the axil of a leaf comparable to the stem tips. So the auxiliary bud which are found in the axil of the leaf, they can be compared with the stem tips. They have meristematic regions. They have the ability to proliferate into new plants by the process of micropropagation. So here what we do in the node culture, we take the nodal segment along with that auxiliary bud and we culture them to develop into a plantlet. Okay. Now closed buds are ideal for micropropagation to reduce the chance of infection because open buds there is a scope of entry of the pathogens, right? That's why we will use closed buds, closed auxiliary buds should be used. They are preferred for micropropagation process. So this is a diagram of your single node technique. So you can see the node has been taken. You all know these, the rounded shown the auxiliary bud. At the tip is a shoot tip. We will not take them. We have already discussed the meristem culture. So this nodal segment is taken and look like this. You can see having the nodal segment, internode segment along with the auxiliary bud. Close auxiliary bud should be used and not fully explanation has been given. Shoot development, you know what we will use. We have to change the composition of culture medium. Low auxin, high cytokinin leads to shoot proliferation. For rooting, opposite of that composition, high auxin and low cytokinin in a suitable ratio. Now that depends on at what ratio of auxin and cytokinin leads to shoot formation and root formation. That's your main objective in a research in plant tissue culture technique. Just like I have done in my MSc, I have done my strawberry culture, and by looking into the research papers, I have they have says that different type of composition, they have used different type of chemicals, phytohormones at different ratios for the growth of shoots and roots in strawberry. And I have also, by looking into the research paper, I have tried to change the composition and found it's good that I can in a particular composition which is different from their composition, ability to grow the strawberry plant in the same way. We These type of research papers normally occur for various plants, like some commercially important plant like for example neem, then shishu, dalbergia shishu, then your normally generally we also go for tobacco plants is the initial part, the basic part where we all try to do the micropropagation process. Okay, so by changing the composition of the culture medium, that which is a suitable ratio of auxin cytokinin that proliferate the shoots and roots, that has to be observed. Okay, next is your axillary bud culture. Now in axillary bud culture, and a nodal culture is not different from them. In nodal culture we are taking the axillary bud along with the nodal segment, but in axillary bud culture we are only and only taking the axillary bud and not extra part of the plant. So both of them are similar, but axillary bud is more, you can say, uh, more fine part of the plant, that we are only and only we are taking the bud, not the nodal segment, right? But in nodal culture we are taking the nodes, internodes little bit, and the axillary bud. So in axillary bud culture, shoot tips along with axillary bud is isolated. Okay, shoot tips along with axillary buds are isolated. The cultures are carried out in high cytokinin concentration. So directly we are taking the shoot tips having that axillary bud, just that part, minimizing the nodal segment, and then we will use them for inoculation in the culture medium with high cytokinin concentration. As a result of these, apical dominance stops. Apical dominance means the shoot apex will stop their dominancy. It will not grow. It will help the axillary bud to proliferate. Schematic presentation has been shown that how axillary buds are used here for the growth of the plant. So these are rosette plant where we are taking only the axillary bud. Okay, and culture, shoot development occurs, root development occurs, and finally we will plant them in soil. This is the part of the plant where we are taking the tips as well as the axillary bud, and we are going for frequent subcultures for shooting and rooting of the culture medium. So this was all about the micropropagation process. Now there is also other two parts which I have not mentioned with you, but it's run out of time. The organogenesis part, which I will do in the next video, and the somatic embryogenesis. Okay, we are now trying to say about the advantage and disadvantage of micropropagation. So I am skipping the pages now. So what are the advantage and disadvantage of micropropagation? Although I know we all know the advantage, first is high rate of plant propagation. Through micropropagation, large number of plants can be grown from a plant tissue within a short period of time and space, irrespective of seasonal variation. So, what season it may be, you can grow the plants in a large quantity. Good commercialization, good business approach, which will make you a lot of money. Second is production of disease-free plants. It is possible to produce disease-free plants by using the meristem tip cultures, or the auxiliary bud can also be used for production of disease-free plants. Why we can produce disease-free plants, I have already discussed with you repeatedly two times, that these meristem tips are actually a site of cell proliferation where the cells are constantly dividing. They are metabolically active cells, so there is no scope for entry of the virus or other pathogens to enter. Secondly, these meristem regions are lacking the xylem and phloem vessels. The conducting tissues are lacking, so it is also a pathway for entry of the pathogens, which is absent in meristematic regions, in meristem regions. Some examples has been given. We have prepared a sweet potato free from virus using tissue culture techniques, which is Epomia batatas, cassava plant, manihot, esculenta, and yam plant, Discoria rotundata. Second, third advantage is that production of seeds in some crops. Now there are some crops which not produce seeds actually, like cauliflower, like onion. So we have to take the vegetative part of the plant and use them as seed. For that, we can go for micropropagation process. Okay, so micropropagation through axillary bud proliferation method is suitable for seed production. It is suitable for seed production in some plant, and this is required in certain plants where limitation of seed production is high degree of genetic conservation. Then fourth is cost effective process. Although micropropagation is not cost effective, it is costly. If you see the equipments that are being used for the micropropagation, even if you are using the laminar airflow, it should be of high safety laminar airflow, not the simple laminar airflow. There is four safety levels of laminar airflow. Okay, so it falls under the number three safety level of laminar airflow, where we are doing the inoculating the cultures aseptically introducing to the culture medium. It is very very costly, so one time investment, but it is very costly. Then all the glass vessels, vials, the media that we are using for tissue culture are also expensive. But why they are saying cost effective, it is the labor. Actually the labor cost here is low. The wages, the wages that we normally give to the labor here is low. Next after China is India, where the manpower and labor charges are very very low. But if you go to UK, or if you go to America, in USA, in Canada, or in South Africa, there the labor charge is quite high, 20 times higher than this charge actually here. That's why they are saying that micropropagation is a cost effective process, right? Then says automated micropropagation. Nowadays, scientists are trying to minimize the manpower and increase the efficiency of micropropagation by using your bioreactors. Bioreactors are specially designed reactors where we can use the liquid medium to grow the callus, which is the suspension culture. Properly agitation or mixing of the culture medium will occur, and instead of using workers, we can use artificial intelligence, we can use robots in place of laborers for micropropagation, which will further reduce the cost of plants that we are growing the plants through micropropagation process. But obviously all the things have disadvantage also, good things also having disadvantage. Like first is contamination of the culture. If you see, when you are doing the culture in further in your MSc classes, you will see, if you have your subject in this my plant tissue culture as practical, you will see that repeatedly culturing, sometimes some little bit mistake can leads to contamination of the cultures. Mainly speaking, pathogens like fungi normally enter to this culture, and your whole uh, labor gets wasted actually. Okay, so during the course of micropropagation, several slow growing microorganisms, they contaminate the culture and they grow. And to control this, what we can use, just like microbiology, we can use antibiotic medium so that the culture is not getting contaminated. But if we give antibiotics or fungicides, it will eliminate the growth of virus, it will eliminate the growth of bacteria, fungi, okay, but at the same time it will also affect the growth of the explants. So that's why we try to avoid giving fungicides. Okay, if the explants are getting affected, then we can give fungicides. We can place a dilute antibiotic solution initially. But during subcultures, if we add fungicides in the culture medium, it can adverse the growth of the propagules, it will adverse the growth of the cultures. Next is brewing of the medium. Now, this is commonly you will see in the culture medium lab, that sometimes browning of the culture medium occurs. And this occurs due to accumulation of the phenolic compounds. Phenolic compounds are normally used by the plants naturally to provide protection against pathogens. But when these phenolic compounds came exposed with the oxygen and with the help of enzyme phenolic oxidase, then it produces a brown color product, which are the inhibitory substance for the growth of the plants. And this brewing of the culture medium can be prevented by addition of some antioxidants like ascorbic acids or citric acid or polyvinyl pyrrolidone to the medium. Sometimes we have to take the care when we are doing the culture, like for example when we are cutting the explants from the mother plant, we immediately have to pour that explant's exposed part, the cut part, into the water so that phenolic oxidation does not occur. So this type of things we have to take care, these small things, otherwise it will lead to big problem. Next is genetic variability. I have already said that when micropropagation is carried out through shoot tip cultures, genetic variability is very low, because shoot tip cultures, they undergo normal mitosis. Okay, the shoot tip cultures contain the meristematic parts, and they show less degree of genetic variability, because the cells of meristematic part of the explant, they divide by normal mitosis and the cells are maintained at uniformly diploid level. But the cells of non-meristematic explants, which are the derivatives of the meristematic explant, they don't divide by normal mitosis, but they undergo DNA duplication, they undergo endoreduplication, where cytokinesis does not occur, no cell division occurs, but still DNA duplication, DNA replication is occurring. So more and more copies of DNA within the cell, that leads to the formation of chromosome having four chromatids, which are called as diplochromosomes. Then chromosomes having eight chromatids, also called as quadruplochromosomes, and polytene conditions of chromosomes occur, which also called as polychromosomes. Okay, so that's why we have to take genetic variability occurs. And why occurs, we have said that we have to take the shoot tip. Okay, good, but it is saying that larger explants are ideal for growth of the plants, but at the same time if you are taking larger tissues, there is a chance of using the non-meristematic parts, so there will be genetic variation. Okay, so that's the thing that we face problem. Another important thing that normally occurs, you will see this type of thing quite often, the vitrification part. During multiple subculture or during repeated in vitro shoot multiplication, you will see that the shoots are becoming translucent, almostly translucent leaves are forming and just look like water soaked. So that type of condition is called vitrification. So during repeated subcultures, you will see the shoots are becoming translucent, just like a glass, shield of glass, and they exhibit just like water soaked, just like they excessively they soak the water. And this type of shoots which are translucent, having translucent leaves, they will not survive for a long time. Such type of shoots can not grow and die, and this is known as vitrification. Vitrification can be increased, can be prevented by increasing high concentration of oxygen in the medium from zero point 0.6 to 1%. So if I increase the agar concentration in the culture medium, then we can minimize the vitrification phenomenon. But at the same time, more agar concentration increase means more solidifying the medium. Thus, the explants will be finding difficult to absorb the nutrients found in the culture medium. So it will reduce the growth rate of tissues. So everything has advantage and disadvantage that I have said. Next is the cost factor. Cost factor, as I have said, that so much of equipments, expensive equipments we are using. Like for example, if you are doing a simple micropropagation technique, we have to use an autoclave, a highly modified autoclave. Although in your schools, in your academics, you will see the autoclave that is used for microbiology, same autoclave is used for the plant tissue culture. That should be avoided, because you know very well, okay, microbiology is used for culturing the bacteria, whereas in autoclave we are here concerning of free of sterilization. Then the laminar airflow that we are using, it should be different from other classes, because in microbiology we are culturing the bacteria in aseptic conditions, and if we are using the same laminar airflow for plant tissue culture, there is a chance of contamination more. Although same laminar airflow can be used, but with a very precaution has to be taken. We have to sterilize them. You have to expose them in UV light for about 1 hour, for 30 minutes, sometimes minimum 30 minutes. This type of things are used, so you can see so much costly factor are there. Then the culture rooms that we are using, I have said now that the culture rooms are maintained at 20 to 28 degrees Celsius with 40 to 50% humidity, and the light photo period of 2000 to 3000 lux intensity for 16-hour photo period. So such type of automated system is done by some sensor. So we have to make the we have to make that room, culture room, that type of thing. Like we have to use sensor systems so that automatically it will decrease and increase the temperature, the requirement temperature for the plant. Okay, they're just not like putting them in a fan or AC will help them to grow, but at the same time it should be closed, it should not be ventilated. And all the tissue culture, plant tissue culture things that we are doing, it should be done in closed rooms. And culture room should be, obviously it should be enclosed. Okay, it should not have any ventilation system. Automated sensor system should be present, so that they can able to regulate the temperature as well as they can regulate the humidity, and at the same time they will maintain the light phase and dark phase, just like day and night. Okay, so much of things. And the manpower. Okay, like for example, sterilizing the medium is conducting by two labors. Then pouring the culture into the vessel is done by some people. Okay, so such type of manpower is needed. At the same time, expensive instruments are needed. So this limited the use of the micropropagation process actually. So we are trying to achieve overcome these problems by reducing the cost factor, using automated systems, one time investment. Okay, for minimizing the cost of plantlets that are generated through micropropagation process. So that's all for this lecture. In the next lecture I will try to discuss about the organogenesis and the somatic embryogenesis process. I hope you have understood this part. If you have any questions regarding this chapter after studying all these things, then please you can ask in the WhatsApp group. I will try to explain the answer. Okay.